DPR, Department of Pesticide Regulation, is a very complex organization, but we're really a science-based organization. I'm really excited. We have 3 incredible scientists today that are going to describe what they do as medical toxicologists and how they help our program sort of figure out how to manage pesticide risk as pesticides are used in society.
So the Human Health Assessment Branch, we do risk assessment. We also, we're divided into 3 programs, just as a quick overview. We have a data review section that reviews toxicology and pharmacokinetic and data that can impact human health that is submitted for pesticide registrations. And then they identify any potential human health impacts and report those. That information then for chemicals that are getting assessed for risk, those go into the risk and exposure assessment sections. Those are the 2 sections that deal with risk assessment. The risk assessment section will take all the information on all the toxicity studies, and they will determine doses that below which you can, the evidence suggests that we don't have any human health impacts. And then they compare these ultimately to the exposures that the exposure assessment section calculates to determine risk. And so that's the basic description that we're going to be talking about today. DPR's mission is you can find this on our webpage. As soon as you log into cdpr.ca.gov, this is the first thing you see on the big banner across the top. And it says, our mission is to protect human health and the environment by regulating pesticide sales and use and by fostering reduced risk pest management. Our human health risk assessment, that's what our section is devoted to, is that portion of the mission. So how do we do that? So very simply, what we need to do to protect human health. We determine when the pesticides are used, what sort of impacts can occur, and then the circumstances when those can happen. And so that's the combination of exposure and toxicity that we'll be talking about today. And then the department overall can then take measures when we identify risks that are of concern. The department can then take measures to reduce those, to decrease exposures that lead to risks. And so the way that we do this may very well require changes in how the pesticides are used. Next slide. And so what is risk assessment? It's basically the process that we use. We do a quantitative risk assessment where we calculate numbers and using the best available science that we have. The thing about risk assessment is that basically the department overall has to make decisions. We have to decide, is it okay to use a pesticide? We can't, unlike folks who are doing research, we can't say we don't have enough information, so wait, because that in itself is a decision. So the department has to make decisions, and that's the position that regulatory agencies are in, is we make decisions with the best information we have, and often that information isn't as complete as we'd like. But this is what we're doing. And so what the risk assessment does is we take information about toxicity from studies that registrants are required to submit and information that's available in the public literature, and we compare that to exposure, estimates of exposure. And basically we're looking for the effects where the doses at which no effects are seen, and then it will add safety or uncertainty factors as appropriate. So we'll move forward with that. And we won't, unfortunately, get into that very much today. That could be a whole talk on itself. So when is risk assessment done? Before a pesticide is registered, that's when our data review section gets involved. And as I was saying, and they're actually looking to make sure that we have a complete database of toxicity studies that are required by law. And they go and evaluate those studies, and they look for any sort of adverse health effects. Are the studies showing anything like that? And then there are times when, before a pesticide is registered or a new use is registered, that we will do a formal risk assessment. Certain circumstances where we do that. And for pesticides that are already in use, we will go back through and do risk assessments. And so that's a lot of what we're doing these days. And then anytime we get information that suggests that there's a potential for risk that we haven't been aware of before, it could be reports of illnesses, it could be new studies coming in. or a new use. We will do risk assessment then. And California is unique in that we do have this state risk assessment program in support of the department's regulatory decisions. Next slide. So the goal of risk assessment is we want to be realistic and health protective. Now the health protective, we want to protect individuals from any sort of injury when pesticides are properly used. And so we focus on when pesticides are properly used, and if they're improperly used, then we have an entire different program that will deal with that, and that's over in enforcement. And we balance, what we need to do also though is to balance those protective assumptions in our risk estimates with whatever information that we have to make it as realistic as we can also. Because if we go too far in the health protective and we end up with everything being an extreme risk, and we may end up, well, first of all, we lose the ability to tell what are the most hazardous pesticides, and we may end up focusing on something that actually isn't the most hazardous, and we also may have the end up canceling uses of pesticide when it really wasn't as hazardous as we might have thought. So how are people exposed? And their next talk here is going to be on exposure, but basically people can be exposed in two ways. When pesticide is being used, the person who's using the pesticide, or somebody who's nearby, or if there's airborne pesticide residues and that can drift off site. Then people can also be exposed to pesticides residues that settle on crops or on treated surfaces or on eating foods that have been crops that have been treated, or in drinking water in some cases. And those are the sorts of things that we look at in our risk assessments. The sources for our data generally, we have, again, pesticide registrants are required to submit data, a large number of toxicity studies for any new pesticide. We also go in and do literature searches for any sort of papers that have been published by universities or institutes. And we will also sometimes conduct exposure or environmental residue kinds of sampling, air monitoring, for example. So the studies that registrants are required to submit can include toxicity, also chemistry, environmental things like that. And the exposure studies are not necessarily required. We do have sources of information for those. Go ahead and. But as we move forward into the 21st century, we're going from the older technology to the newer technology. A lot of the studies that are the required studies that have been used for decades, they're approaches that were developed in the 90s, 70s, 80s, and 90s, and sometimes even earlier. And there are newer types of studies out there to avoid animal use.
So what is a human health risk assessment? Basically, it estimates the risk to humans from exposure to pesticides. Now, the key point here is it is based on scientific data, and I will be referring to this again and again and again. This is based on scientific data. It predicts the type of health effects. For example, a pesticide may affect several organs, a lung, a liver, brain. It may affect the whole system, endocrine, reproductive, developmental state of developing organisms. And we also, the risk assessment predicts the magnitude of the effects. In an ideal situation, a scientist, a risk assessor, should be able to exactly estimate the risk from pesticide exposure to humans. In reality, this is never the case. And the reason is we don't have enough scientific data. Because of lack of data, in many cases, we are forced to use conventions, extrapolations, or uncertainties. For example, if a pesticide affects lung and the study is done in animals, we have to have sufficient information how different the lung in animal is from the lung in humans and extrapolate the dose. If we don't know enough, we just can't estimate the risk precisely. And so here is a statement that I want you to remember. The toxicologically based risk assessments are valid within their assumptions. Today we can estimate a risk which we are confident based on the Science today. A year from now, as new science come, we can change our risk. Require the department to determine the adverse effects to pesticides through the risk assessment process. Around the same time, in 1983, the National Academy of Sciences, and I'll stop here for a second to tell you about NAS, or National Academy of Sciences. This is an organization that was created in 1863 by President Lincoln. Best scientists were gathered together to advise the nation on scientific issues. So in 1983, the National Academy of Sciences came with a framework called the four-step process of risk assessment. And here are the 4 steps. Hazard identification, the dose response assessment, the exposure assessment, and the risk characterization. And since I'm in the risk assessment section, I'll be talking what we do in our section. So typically, the DPR risk assessments are about 100 to 200 pages long. And in the risk assessment section, we do 3 of the major steps in the 4-step process that NAS identified. We do the hazard identification, dose response evaluation, and the risk characterization. The exposure assessment is done in the exposure assessment section. So, remember the 4 steps? The first one is hazard identification. This is about half of the risk assessment, and this is where the toxicology is evaluated. Basically, we develop the profile. We get to know the chemical, what it does, what type of effects we see in animals and humans and other organisms. Again, this is based on science, so we summarize all available studies. Big part of our studies are those that Sheryl talked about, the registrant submitted studies. They follow FIFRA guidelines, the Federal Fungicide, Insecticide, and Rodenticide Act. In addition to those, we review all available published studies that are relevant. They do not follow specific guidelines. And here, I have this picture now. Two of us. These are scientists, a few of them reviewing studies from our branch. And so, you can see those are called data volumes. You can see them in the library. For any given pesticide, we may get hundreds of those, so we have to review them, and we often get together to discuss them. Those are heated discussions. Sometimes feelings are hurt, sometimes consensus is not made, but at the end we come together. So we've covered the hazard identification now. We know the chemical, what it does, the type of effects now, we have to quantify those. So this is called the dose response evaluation, the second step in the risk assessment process. Basically, the effects can be divided into 2 types, the non-cancer effect and tumors. So for non-cancer effects, the goal from a scientific study that we review is to identify a dose called no observed effect level, or NOEL. I'll be talking about NOEL, so please remember this. The NOEL is the dose that we do not expect to see a health impact. At this dose or below, anything above is likely to cause some effect. For cancer, NOELs are not calculated, but rather potency is estimated based on tumor incidence in animal studies. So to do this dose response estimation, various approaches are used, and mathematical models, for example, our software that we use is benchmark dose analysis. We use it to calculate NOELs or to estimate potency. And so here is a table that summarizes the NOELs. Remember, this is the dose that we do not expect to cause health impact. Various studies are listed, and you can see different species, rat, rabbit, mouse. The duration is short, acute or short, from 1 day to 14 days. And here in this column, various effects are listed. Those are the most sensitive effects that we've seen in the study. In this case, it's an enzyme that's inhibited in the brain. In this case, in the mouse, the body weight is decreased, like 10% body weight is a very significant decrease. And then in the rabbits, those are pregnant animals, maternal as well as fetal effects are seen. And so we looked at these NOELs, and then we select the lowest one. This would be the one that might be used as a regulatory endpoint. And then we determined that rat is the most sensitive species based on this table. This is a benchmark dose analysis that we put into the dose response evaluation section. So basically, it's a mathematical model. Those are experimental points, and it's a curve fitting program. And here you can see those two doses, somewhere around this area is the dose that we do not expect to see health impact. And so I covered the first three parts of the risk assessment process, and now the exposure assessment is for Eric.
Consider actually is where this happened and how the chemical, in this case a pesticide, enter into the environment, and how the exposure occur, meaning is it through oral, for inhalation, or through dermals. For the exposure factor, that I will elaborate a little bit, but for now it's practically mean that the inhalation rate or the body weight of the population are being exposed, and that tied to the like some other thing like the activity pattern, how much time you spend in an area or at a home. This is the actual application for methylbromide. Methylbromide actually has a boiling point around 3.5 degrees Celsius. So what that means actually, under the ambient condition, this is a gas. So although the chemical directly inject into the soil, so because it is a gas, it will actually come out from the soil and get dispersed to its surrounding area. Now, because of that, we usually set up the monitoring station around the field to try to monitor the actual air concentration of a pesticide. This is an actual diagrammatic representation of an air monitoring study conducted in Turlock County. Now, although this is not a soil fumigation process, but the whole idea is that during the application process, as Sheryl described, the pesticide will get actually moved off-site to the surrounding area. This diagram shows the actual monitoring set up by DPR to monitor the air concentration of pesticide in the air. Now, the monitoring data are important, but are not necessarily adequate in terms of capture the pesticide concentration, and the next slide will demonstrate why. This diagram shows the movement of the pesticide from the field. When the pesticide volatilized into the air, it got mixed, and then it carried by the wind. This model is characterized by so-called the calcium plume model. So what they really mean actually is the concentration kind of like peak at the top, and then it kind of like get decreased on the both sides. ICST3 model has been used by DPR as a pioneer model for characterizing the air concentration of pesticide. Now, this is another example of the air dispersion model, but it's a slightly complex. This is called the soil fumigant exposure assessment system, or called SOFIA. The SOFIA model actually has a characteristic add-on to the ISCS-T3 model. What it does is allow actually to use the field size, the application rate, and other characteristics like the land use to realistically characterize the pesticide concentration immediate or escape from the application site. And this flow chart is simply a kind of like I'll show you actually what the SOFIA system. The main computation engine of the SOFIA is still the ISTS3 model. But as I mentioned earlier, the program also allows you to put in the actual use practice. So how often you apply the pesticide and what are the land use, what the acreage. So in that way, it will give you a much more precise or more realistic determination of the actual pesticide use. And because of this information, when you go to the ISCS-T3 model, the model outcome will be more closer to the actual pesticide concentration that you would observe in the air. This is a contour plot of the pesticide concentration predicted by the ISTS-T3 model. As you can see, these are the color coding of the concentration. The higher concentration are observed in the 4 upper right townships. These townships also have a higher 1,3-D use than the other townships. The purple number represents the actual monitoring data. So by comparing the monitoring data to the predicted air concentration, we can use it as a tool to check or to validate the model, so that when we apply the model for the human risk assessment or exposure assessment, we'll be much more confident. This slide shows a closer look of the monitoring versus the predicted air concentration. The dashed line represents a perfect correlation, meaning the predicted versus the measure will be exactly the same. Anything above will be an overestimation, under prediction. As you can see, most of the time, the model actually overpredict the concentration, sometimes it underpredict, and we are in the process of continue evaluating this model so that we will ensure that the underprediction occur as minimal as possible. Remember I showed you earlier the exposure equation consists of two factors. One is the air concentration, the other is the exposure factor. The exposure factor are associated with who are being exposed. And for the purpose of this presentation, I would like to use the general population as an example. And that we call it a bystander, meaning the person who are not actually involved in the pesticide application process. As you realize, the population is rather diverse. It consists of infant, children, and adults. Without going into too much of the detail, these two models are so-called the stochastic model. What they really mean is that it randomly selects the individual from the entire population, and then to calculate the exposure. Now, because different individuals are selected, instead of you getting a single exposure value, you get a distribution or range of exposure value. And that's what the Monte Carlo distribution about. For those of you who love mathematical equation, I just show you a little bit more detail about what the model actually looked like. Yeah. So as you can see, actually, the equation consists of the air concentration, meaning the one that we model at the different townships. And also we consider the breathing rate because a child has a slightly different breathing rate than an adult, and so has the body weight. So again, we put all this information together and then to estimate the exposure for the entire population.
Now the last part is called the risk characterization. So this is the final step, and it is used by the risk manager to develop control or mitigation strategies. What we prepare is eventually going to the managers to decide what to do with the pesticide. So the important part is the risk characterization uses exposure, the estimates that Eric's group calculates, and the dose response assessment that we prepare in the risk assessment section. So remember how I told you basically the effects can be divided into non-cancer and cancer. So the risk characterization calculates the risk. For effects other than cancer, the risk is typically estimated as in terms of MOEs or margin of exposures. So this is exposure and dose response. Noel is the dose that I showed you in the table earlier, that we do not expect to see adverse effects in animals or humans, and the exposure is estimated. It's a human exposure estimate. So this is a number. By itself, it means very little. We can get from 1 to thousands. What's important is that it's compared to a target MOE. So the target MOE, we recommend the target MOE. It is usually of 100 when data comes from animal studies. So this completed the 4 steps in the risk assessment: the hazard identification, the dose response evaluation, the exposure assessment, and the risk characterization. Now we calculated the risk. We have to assess it. We have to tell the manager how confident we are in the numbers. So I want to talk to you about the studies that we used, because the studies are those that makes the risk realistic or not realistic, or makes us add more or less uncertainties. So ideally, we should be using human studies. This is a human health risk assessment. Unfortunately, they are rarely available. We can't take humans and dose them in the lab to see. So there are some done, and if we find a human study that's available, DPR would prefer that it has been approved by the Human Studies Review Board. There are other kinds of studies that are not specifically designed for risk assessment, but they're done in humans. They're not done, they are just available, or some of them are done. And so those are epidemiological studies, human case reports, or human illness reports. In some cases, those are deliberate, they are poisoning, self-poisoning, or accidental. They're very informative. They tell us what kind of effects we would expect to see in humans if they're exposed to this pesticide. But in most cases, the effects are either self-reported or they're not evaluated by physicians, and we do not know the exposure. So we cannot use them to calculate risk. So when we don't have studies in humans, we use animal studies. And so Sheryl touched a little bit on the animal studies. They're required for pesticides to be registered in the United States and in California. And usually, and the requirement is that the animals are dosed through the oral, dermal, and inhalation routes. These are the routes that people can be exposed. We can eat food that's contaminated with pesticide residues. We can get exposed through the skin, or we can inhale air that has pesticide residues. Multiple laboratory animal species are used. That is required by FIFRA. If you remember the table that I showed you, there was a mouse, a rat, a rabbit study, and we typically receive studies in dogs as well. What we do is we look for the most sensitive animal. In the table that I showed you, it was a rat, and at the lowest dose at which health effects appear. And of course, humans are not animals, and there is an uncertainty for extrapolating the findings from animals to humans. So that's the key question. What do we do with animal studies, and how do we extrapolate them to estimate the risk in humans? So there are various ways to do this, and it's called scaling of the dose among and within species. So we can scale the dose within species between rats and humans, or within a species. For example, we can scale the dose between an adult and a child or an infant, or we can scale the dose between different status in humans, for example, non-pregnant versus pregnant adults. So one way to do it is called the body weight based scaling. Basically, we extrapolate the animal dose to the equivalent human dose based on the differences in body weight. Another way of doing it is called target tissue based scaling. This is a newer approach. It uses new science, and it extrapolates the dose in animal that is found at the target organ, for example, the lung, to what the dose would be in human at the human lung. And we're very proud to report that we employed a target tissue scaling called regional gas dose ratio, RGDR, in one of our most recent fumigant risk assessments. We have our lead toxicologist, Dr. Rubin, here. And is Chuck here? Chuck Aldous is the lead of the data review, and Eric and Ian are the leads of the exposure assessment. And so I want to demonstrate this in this slide, how we extrapolate. So an adult human, a child, and a rat, and you can see there is difference in the body weight, so we can scale based on the body weight. Or we can use the lung, using the knowledge that we have for lung in rat, to what is the difference between the lung between human and rats, or between adult and young child, to scale the dose. Okay, so we're looking into new ways of testing pesticides and environmental chemicals. And so in 2007, the National Academy of Sciences published a report on the future of toxicity testing, known as Science and Decision and Toxicology in the 21st Century, Tox 21. So this is a federal program that involves government, universities, and industry, and the goal is to develop better toxicity methods to reduce the animal testing. They typically focus on mechanisms of toxicity. For example, if we take one animal study that takes 2 years, the lifetime of a rat, we would have to wait until the last month to assess tumors in the animal. However, so it takes 2 years, and it's an expensive, long process. And if it doesn't work, the study doesn't work for a number of reasons. For example, control animals die from contamination, totally unrelated to the chemicals, then it has to be done again. Now, the mechanism, if we know the mechanisms, we can predict whether an animal would develop, or a person, a human, would develop tumors based on the very early indications, very early in the treatment. So we're looking for signatures or biomarkers that would indicate what will happen later on. Instead of animals, cell cultures, biochemical reactions are used in vitro. And then those methods are automated. A large number of chemicals can be tested, and many tests can be run in one place. And now, we're super proud of those 3 risk assessments. This is the one for 1,3-dichloropropene, which was finalized in December, and it's available on the web. It took a lot of efforts and a whole work group to work on it, but we incorporated this new type of scaling, not the body weight based scaling, but the target organ dose scaling, the RGDR. And this one underwent peer review by OIHA as well as the University of California. So it's been extensively reviewed, and those 2 are still drafts. They're available on the DPR website. And the reason they're drafts, they were completed in December, but they're still undergoing external peer review. One is for chlorpyrifos, the other one is for dicrodifos. Those 2 incorporate the new Tox 21 approaches. We used a particular method called ToxCast, which predicts mode of actions. And so that's my last slide, and I'll summarize the talk is that new techniques are promising, there is a lot of work, and what we're trying to do is validate the new studies, the in vitro studies, with what we know from the old established long-term animal studies. And here is the trivia question. What is this organ? Yeah. Okay, and here is a quote that I learned yesterday from Andy. And risk assessment is easy. You can learn it in 2 steps, and each step takes 10 years.